Hi class, so it's time to continue on with our anterolateral pathway. And the anterolateral pathway is also known as the spinothalamic pathway. And this is going to consist of anterior and lateral white funiculi. Don't let the word funiculus confuse you. It simply means a collection of axons inside the central nervous system. Now if you look carefully at this picture, go all the way down to the bottom of the picture. You're seeing that we have a three-neuron chain. We have the purple neuron shown here at the bottom of the picture, synapsing on two different slightly colored blue neurons whose axons ascend all the way up to the thalamus, where they synapse on green neurons that are found in the thalamus, whose axons then are going to terminate ultimately on the primary somatosensory cortex. The anterolateral pathway is primarily concerned with pain and temperature, which is the primary focus of this lecture, of course. So, by the words anterior and lateral, we are simply meaning that there is a tract of white matter. And remember, white matter means axons that are myelinated, that are ascending up to the brain from the anterior portion of the spinal cord and the lateral portion of the spinal cord. So if you look back down at the very bottom of this picture where it says pathway direction, you see an arrow, and it's pointing to a little sliver of the spinal cord. You'll see that there is a tract on the anterior portion of the spinal cord, and then the slightly darker blue tract is slightly over to the side, right? So that's the lateral portion. And forgive me, but these terms are really, really technical, and you probably don't remember them from anatomy, which is why I'm saying them again to you and hoping that your memory recall will be faster than when you learned these pathways the first time when you were in anatomy. So how's the anterolateral pathway different than the posterior funiculus medial lemniscal pathway, which, boy, is that a mouthful of words to say. It's similar, first of all, in that it has a three-neuron chain, which so did the posterior funiculus medial lemniscal pathway did. But it's different. If you look very carefully down at the bottom of the picture, you'll notice that we're having crossing over, decussation, at the level of the spinal cord. It's not up in the medulla like we saw in the posterior funiculus medial lemniscal pathway, which is for discriminative touch. So that's a big difference here. We're having crossover at the level of wherever the pain and temperature is being relayed from. The anterolateral pathway is also important for crude touch and pressure, but for our intensive purposes, we're going to focus on pain and temperature. Now, where the secondary neuron synapses on the tertiary neuron, or third order neuron, it's up here at the thalamus. Move your eyes upward and in our picture, and you'll see that the blue axons are terminating on the green neurons. Now, where this termination occurs, the synapse, this is where our first realization of pain is going to occur. But you don't know exactly where that pain is coming from. You just know that you're hurting. You can't really localize it. And of course, pain, in order for it to be protective or effective, it needs to be localized so you know if you have pain in your foot, you should move your foot. If you have pain in your arm, you should move your arm away from that painful stimulus. So that's the protective effect of pain. But you don't get that awareness of specificity when you have the second-ordered neuron synapsing on the third-ordered neuron in the thalamus. It's not until the third-ordered neuron terminates in the primary somatosensory cortex that you get the idea of where the pain has originated from. And I'll remind you from our first segment, thanks to the review of the homunculus, we know that the primary somatosensory cortex is designated to serve the needs of different body regions. And that's already been mapped. That's called somatotropy. And we also learned from our first segment that the larger the picture that was portrayed on the primary somatocortex, primary somatosensory cortex, that means more neurons are dedicated to serving that region of the body and interpreting the signals from that region of the body. And I made a decently crude joke about the size of our genitalia on that somatotropy map. I hope you share my humor. So what should you know about the anterolateral spinothalamic tract, as it's called? You should know it conveys pain and temperature. It has 3 neurons. You should know that the primary neuron, or as we say, sorry, I'm losing my voice here. The first order neuron is going to come in from the periphery and terminate on the posterior horn. This is where the second neuronal cell body is found. And the second neuronal cell body, the axon from it will immediately desiccate, cross over to the opposite side, again contralateral, but this crossing over occurs much, much earlier, meaning more inferior on the spinal cord than the posterior funiculus medial lemniscal pathway that we saw in the previous segment. The third ordered neuron, or tertiary neuron, is found in the thalamus, and it's going to project onto the primary somatosensory cortex. Now, in the next few pictures, we're going to get pretty technical. And the reason why we're going to get pretty technical is because I have to explain to you that afferent fibers, incoming sensory conveying fibers, and the word fiber is a fancy way of saying axon. So I'm going to have to tell you in the next few slides how we classify afferent fibers. They're either A or B or C fibers. And then A fibers have further classification. So I need to do that in the next slide. And then the slide after that, I have to tell you more about the posterior or dorsal gray horn. That's the portion of the gray matter in the spinal cord. That would be, if you're thinking of a butterfly wing when you're looking at these pictures of the spinal cord, the tops of the butterfly wings, not the bottoms, the ventral gray horns. So the dorsal gray horns are mapped, or according to anatomists, they're arranged in layers. And the word for an anatomist that means layer is lamina, and plural is laminae. So again, to warn you, it's going to get pretty technical with the vocabulary, but I'm going to try and stop and say, okay, what does this mean? What should you be focusing on? Okay, I'm going to spend quite some time on this picture, and it seems so straightforward, but I want to make sure you're following along. First of all, look at this picture in terms of rows. At the very top, you should read that there is diameter, and that means the diameter of the axon for each of these types of neurons. So from the left side, we're going from very large to very small on the right side. And just to remind you that the larger the diameter, the faster the rate of conduction for the action potential. You can think of this in the following way. If a freeway is very large, such that you have 8 lanes in either direction, you got to think about the flow of traffic being amazing. But if you have a very highly traveled freeway where there's only 2 lanes on either direction, this is going to have high congestion. And so the larger the diameter, the faster the rate of conduction. Now that's the second row you should be seeing here. And if you look on the left side where the fiber is very large, say 20 micrometers, you're seeing a very fast conduction velocity. Whereas when it goes back down to 0.5 micrometers, the very next row is showing you that it's very slow in conduction velocity. Okay, so far so good. Now, if you look in the third row, it's showing you general classification. And it's basically showing you your A classification, your B classification, and your C classification. Now I've jumped down to sensory nerve classification. Now if you look down in your sensory nerve classification, look, you'll see a 1A and a 1B, and then you'll notice that there is the number 4, which corresponds with the general classification above as C. Now, I don't want this to get too crazy. Let's whittle this down, shall we? We are going to be playing with either A fibers or C fibers. Let's just leave it as that. Now, the A fibers, you're noticing, are reduced down to more subcategories: alphas, betas, gammas, deltas. And the C category just stays the same. Okay, well, let's address the C category. Those are just unmyelinated, very small fibers. Now, if you took physiology at Saddleback, you'll remember your PhysioX, where we played with a computer simulation where we played with frog neurons and an earthworm. Do you remember that? And the axons that were the smallest and the unmyelinated had the slowest conduction velocity. All right, so that's what this chart is here to show you. So now let's add another layer to it. Some signals need to be transmitted to or from the central nervous system extremely rapidly. Otherwise, the information would be useless. An example of this is the sensory signals that apprise the brain, tell the brain of the momentary positions of the legs at each fraction of a second during running. It's a good idea to know where your legs are in space while you're running. At the other extreme, some types of sensory information, such as pain, aching pain, do not need to be transmitted rapidly. So slowly conducting fibers will suffice. In this figure, you should be seeing that nerve fibers come in all sizes between 20 micrometers in diameter, very large, to 0.5, very small. The larger the diameter, the greater the conducting velocity. Again, how fast can you get home on an 8-lane freeway versus a 2-lane freeway? The range of conducting velocities is between 0.5 and 120 meters per second. Now let's look at the general classification of nerves. Shown in this figure, under general classification, there is also below that a sensory nerve classification. Since we're talking about ascending tracks, we're talking about sensory nerves. Let's look at that. You'll see, in the general classification, first of all, there are fibers divided into types A and C, okay? And the type A fibers are further subdivided into alpha, beta, gamma, and delta, okay? Great. Type A fibers are the typical large and medium-sized myelinated fibers of spinal nerves. If you look all the way up at the top of this, you're seeing myelinated versus unmyelinated. Type C fibers are the small, unmyelinated nerve fibers that conduct impulses at low velocities. The C fibers constitute more than 1/2 of the sensory fibers in most peripheral nerves. The sizes, velocities of conduction, and functions of the different nerve fiber types are, they are of importance to us. We now want to look below and see the next category that says sensory functions. Please excuse me. A cough is going around in my household and I feel like I'm choking. Now you should be looking at the blue boxes that are down in the sensory functions category. You don't have to go down to motor function because we're not talking about outgoing commands. We're only focusing on relaying incoming information. Now this is what I want you to realize. There's your A alpha, your A beta, your A gamma, and your A delta, and they're getting progressively smaller. If you take that general classification and go down to the sensory function, I want you to realize that when we're talking about muscle stretch or tactile information, like information from our Meissner's corpuscles or Merkel's corpuscles or Messinian corpuscles from our previous lecture, those will all be conveyed through your A alpha and A beta fibers. Large fibers very fast. You know when you're touching something very quickly. Now your deep pressure, like your Ruffini's, that's going to be your A gamma fibers. They're going to be smaller and a little slower. But now look at your A delta. Notice that the A delta go all the way down to where you first start seeing your blue boxes, and you should see that A delta is mostly that pricking pain. This is your fast neospinothalamic pain. The more newer types of tracts to convey pain on the evolutionary scale. Now look at your C classification. Go all the way down, and you'll realize that the C fibers that are smaller and slower, notice they are primarily concerned with conveying information about pain, aching, throbbing pain, temperature, and tickle. And again, I want to remind you that there is a point where temperature can be perceived as pain. So hey, that's our focus for right now. Our A delta fibers that are larger and faster than the C fibers. And our A delta fibers are going to be conveying information about the fast occurring pricking pain, and the C fibers that are unmyelinated are going to be conveying pain that is slower to come to our consciousness and is going to be more longer lasting. So to bring the story back to my initial story about when I burned myself, let me ask you this. Which fibers were initially carrying impulses up toward my brain when I first had the sensation that something was hurting, but I didn't quite know where, and it was very short-lived, versus which fibers finally kicked in and conveyed information, and I felt that throbbing, burning pain. So when I first touched the broiler, those were my A delta fibers, relatively large, myelinated fibers, and I felt that sharp pricking pain, but it was not very remarkable. That's what drew my attention. I didn't quite know where it was, what's going on, and that's where I kind of watched my skin melt and then stick to the broiler. I know it's gross, but it's so true. And then my C fibers that were smaller still and unmyelinated, once those kicked in, that's when the signals reached my thalamus and my primary somatosensory cortex, and that's when I registered that throbbing, really aching pain, and that's when I finally withdrew my hand. So that's our focus for the rest of this lecture. Okay, my sweet class, I'm going to spend a lot of time going through this slide, and I hope my voice lasts for me, but I'm recording this on a day when I had physiology, and it was a big day of lecturing, and I'm kind of losing my voice. So I apologize in advance if I'm being irritating to you. First, I want you to look at the top right picture that's on your screen, and I want you to realize that it's a section through the spinal cord. You're seeing your traditional butterfly shape of the gray matter. And I want you to look on the left side of that top picture, and you will see in a dashed line a circle around two axons. It says spinal nerve. And you'll notice that one of them is labeled C and one is labeled A delta up at the top left of that top picture. Now the C represents those small unmyelinated fibers I just described to you, and the A delta represents those somewhat larger and myelinated fibers I just described to you. Now look at the gray matter that's shown on that picture. You'll notice it has Roman numerals on it, and they're numbered 1 through Roman numeral 9. So this is basically an anatomist putting the gray matter into layers, or lamina. So there's lamina 1 through lamina 9. It just simply means layers. Again, don't let the terminology scare you. The plural for lamina is laminae. Now, if you look carefully in this picture for the C fiber or A delta fiber, you'll see that the primary neuron that's coming into the dorsal gray horn will terminate on different layers or different laminae. And I'm going to talk about where this primary neuron first initially terminates and those different laminae. And again, it's going to get very technical, and I'm going to remind myself to stop and say, here's what I want you to know, or here's what I want you to pay attention to. So let's start first by saying there are two pathways for pain to be sent up, or at least the sensation of pain to be sent up to the brain for interpretation. We know there is the slow pathway, or the paleospinothalamic, and the fast pathway, or neospinothalamic. We're really talking about the anterolateral pathways. The A delta fiber that I just introduced to you is part of the fast pathway, the neospinothalamic. Neo meaning more new, newer in evolutionary time. The C fiber, smaller, unmyelinated, is going to convey information that will be interpreted by your brain as the more burning, throbbing, aching pain. This is the paleospinothalamic pathway. So let me say it again. Even though all pain receptors are free nerve endings, these endings use two separate pathways for transmitting pain signals into the central nervous system. The two pathways mainly correspond to the two types of pain: a fast, sharp pain pathway, neospinothalamic, and a slow, chronic pain pathway, paleospinothalamic. So we have fast and slow. The fast, sharp pain signals are elicited by either mechanical or thermal pain stimuli. That basically means that if you were to ram up against your desk, that is enough to start the pain signals, the action potentials, to be conveyed through the the A delta fibers. But it also can be temperature. Remember, temperature can be interpreted as pain once it gets beyond 45 degrees centigrade. So, because of this double system of pain innervation, a sudden pain stimulus often gives a double pain sensation. What do I mean by that? A fast, sharp pain that is transmitted to the brain by the A delta fiber pathway is followed a second or so later by a slow pain that is transmitted by the C fiber pathway. The sharp pain apprises the person rapidly of a damaging influence, and therefore plays an important role in making the person react immediately to remove him or herself from the stimulus. The slow pain tends to become greater over time,
This sensation eventually produces intolerable pain and makes the person keep trying to relieve the cause of the pain. Again, both of these are going to have protective effects. On entering the spinal cord, the pain signals take two pathways to the brain, either one, the neospinothalamic tract, or two, the paleospinothalamic tract. If we focus on the neospinothalamic tract for fast pain, the fast type of A delta pain fibers transmit mainly mechanical and thermal pain. Again, you just run into your desk or you've touched the candle that's burning. These primary neurons, first-ordered neurons, terminate mainly, if you look at this top right picture, in lamina 1. This is in lamina 1 of the dorsal gray horn. And there they excite second-order neurons of the neospinothalamic tract. These second-order neurons give rise to long fibers that cross immediately to the opposite side of the cord. This is contralateral. This is crossing over. You'll notice that some of these fibers terminate, never mind, I was getting ahead of myself. You'll notice that these second-ordered fibers are crossing over immediately, and they are going to go up to the thalamus and then synapse on third-ordered neurons, which you cannot see in this picture, but you'll see that the axons, the tracks, are circled as fast, sharp pain fibers. So again, there's immediate crossing over, and this is very different than our posterior funiculus medial lemniscal pathway that we learned previously. Now, where do those second-ordered fibers go? If we look at the bottom picture, we can kind of follow them up to the brain. A few fibers of the neospinothalamic tract terminate in the reticular areas of the brainstem. The reticular area is a diffuse area of the brainstem. There's no one specific region. But most of the neurons pass all the way to the thalamus without interruption. And they're going to terminate in the thalamus, right? And we see that up here. Now don't worry about the whole the ventral basal complex, etc. But that's where they're going to go to. And those third-ordered fibers are then going to be transmitted to the somatosensory cortex. Now let's continue on with our story. When the second-order neurons terminated on the thalamus in the example of the neospinothalamic pathway I just told you, there can be a conscious awareness of pain. However, the pain is not localized. It's not until it's projected onto the primary somatosensory cortex that the pain can be exacted. And the next question is, what are the neurotransmitters used in this neospinothalamic pathway? It is believed that glutamate is the neurotransmitter substance secreted in the spinal cord at the level of the A delta pain fiber ending. So where the A delta pain fiber comes in and terminates on the first lamina, it's releasing glutamate, which is the largest widely used excitatory neurotransmitters in the central nervous system. And it usually has a duration of action for about a few milliseconds. But again, this is excitatory. So most certainly our action potentials are going to be conveyed upwards towards the thalamus through the second-order neurons, because they will reach threshold, spike in action potential, and transmit it up to the third-order neurons. Their cell bodies, of course, found in the thalamus. Now let's focus on the slow pain, or the paleospinothalamic pain. Now let's focus on the paleospinothalamic pathway. The paleospinothalamic pathway, like I said, is a much older system and transmits pain from the slow, unmyelinated type C pain fibers. In this pathway, the peripheral fibers terminate, if you notice in the top right picture, in the spinal cord, the dorsal gray horn, almost entirely in laminae 2 and 3. Now, laminae 2 and 3 are together called the substantia gelatinosa. Why would they be called such a thing? So bizarre. They're called that because when they were stained by early anatomists, they stained the darkest of all the 3 layers. And they had kind of a gelatinous look to them when they were stained. So, gelatinous substance means substantia gelatinosa. So that's laminae 2 and 3. Now, most of the signals then pass through another set of short fiber neurons within the dorsal gray horn itself on that side ipsilaterally onto lamina 5. And then from lamina 5, you'll notice, then there's crossing over to the contralateral side where those technically second-order neurons are now going up with the same tract as the fast-ordered neurons, the fast-tracked second-ordered neurons. So again, crossing over occurs at level for the paleospinothalamic pathway. Now, the initial cause of the action potential in the first-ordered neuron is not so much a mechanical start as it is a chemical start for the action potential. And this means that tissue damage has occurred. Now, what chemical signals do tissues release when they become damaged? Bradykinin, potassium, I'll remind you, leaks from the cells. Acetylcholine can even be used here, histamine, and serotonin. Now, bradykinins are really interesting. They've been studied for quite some time. And the reason why they're interesting is because they stimulate the nociceptor. They stimulate the free nerve ending. Bradykinins have been around for a while. Maybe you've heard of a fine young gentleman from Uganda, Idi Amin, back in the early 19th century. Well, he was in power when I was just a young child, but he was well renowned for his torture of the people that he commandeered in Uganda. And he actually had these people tortured by injecting bradykinin into their skin, which caused excruciating pain because, again, it was stimulating that paleospinothalamic pathway. And he thought he was getting away with it because it left no overt marks. A really fine chap, I'm sure you'd appreciate. Anyway, with the C fibers and the slow pain pathway, we're talking about chemical signals released by the damaged tissue leading to the action potentials in that first-ordered neuron. Now let's continue on. Now, from these C pain or type C pain fiber terminals, again, the terminal boutons of those primary or first-order neurons entering the spinal cord, research suggests that they release glutamate, but also it suggests that primarily they're releasing a substance called substance P transmitter, known as just substance P. And P doesn't stand for pain, it's just substance P. Substance P is released much more slowly, building up in concentration over a period of seconds or even minutes. In fact, it's been suggested that the double pain sensation that one feels after a pinprick might result partly from the fact that the glutamate transmitter gives a faster pain sensation, whereas the substance P transmitter gives a more lagging sensation. Regardless of the yet unknown details, it seems clear that glutamate is the neurotransmitter most involved in transmitting fast pain into the central nervous system, and substance P is concerned with the slow, chronic paleospinothalamic pathway. Now, Now let's move on with the projection of the C fibers and the second ordered fibers in the slow pain pathway. The slow chronic paleospinothalamic pathway terminates widely in the brainstem, and only 1/10 to 1/4 of the fibers pass all the way to the thalamus, where they would terminate on third order. Instead, most of the second-ordered neurons will terminate in one of the three areas. Again, warning, it's going to get technical. Either the reticular nuclei of the medulla, pons, and mesencephalon, and I'll remind you that nuclei means a collection of neuronal cell bodies inside the central nervous system. So we're talking the reticular nuclei. Again, the reticular activating system is the system that helps arouse you from sleep, helps you to remove background noise while you are studying. It basically helps you keep alert. And this is found in many regions of the brainstem, but most notably here we're noting that we're going to target the medulla, pons, and mesencephalon is the midbrain. 2. The tectile area of the mesencephalon, deep to the superior and inferior colliculi. Basically, this is the region where your eye reflexes and ear reflexes are going to turn to any loud, threatening object or sound. So when you're in pain, you want to turn to whatever is threatening you. And 3, the periaqueductal gray region. So the periaqueductal gray area, or periaqueductal gray area is sometimes abbreviated as PAG for periaqueductal gray and then area. And this periaqueductal gray area is found surrounding the aqueduct of Sylvius, which is a old and antiquated way of saying the cerebral aqueduct, which connects, of course, the third ventricle to the fourth ventricle, just to remind you of your anatomy. Now again, this slower pain pathway is older in evolutionary terms. These lower regions of the brain are also older in evolutionary terms. There are reptilian brain that you were learning in Paul MacLean's triune brain theory. And if we look at animals whose brains have been sectioned above the midbrain to block pain signals from reaching the cerebrum, these animals, although not consciously aware of the pain, they still show undeniable evidence of suffering when any part of the body is traumatized. So they may not be consciously aware of it, but you, the researcher, could very most certainly tell they are in pain. From the brainstem pain areas, multiple short fiber neurons relay the pain signals upward into the thalamus and then into the hypothalamus and the basal regions of the brain. So what should you be taking home here? This slower pain pathway is older in evolutionary terms. It doesn't necessarily get to the cerebral cortex for conscious interpretation. It is our more and most primitive way of detecting pain. And even when the cerebral cortex is disengaged in animals that have been studied, when they are subjected to trauma, they most certainly are displaying pain. And it's sad that we're doing animal research, I get that, but it's important to know this. And although they're feeling the pain, they are not able to exactly localize the pain and interpret the pain quality. That's what we need the cortex for, and that's a later structure that has evolved in our brain. So to continue on with that, localization of pain transmitted through this paleospinothalamic pathway is imprecise. For instance, slow chronic pain can usually be localized only to a major part of the body, like to one arm or leg, but not to a specific point on the arm or leg. This is in keeping with the multisynaptic diffuse connectivity of this pathway. Again, we're going from the C fibers to lamina 2 and 3 to lamina 5, then we're crossing over, then we're going to the brainstem, the pons, the medulla, the midbrain, then we're going to the area around the cerebral aqueduct, we're going to areas of deeper areas of the midbrain, and then we're moving on to the hypothalamus and the thalamus and basal nuclei of the brain. We're not even really getting to the cerebral cortex. So again, we're not going to, or animals are not going to be able to discern exactly where that pain is coming from. Now, what about these areas of the brain, the reticular formation that I was talking about, and their involvement in the appreciation of pain? Complete removal of these somatosensory areas of the cerebral cortex, again, does not destroy an animal's ability to perceive pain. Therefore, it is likely that pain impulses entering the brain, particularly the brain stem, the reticular formation, the thalamus, the hypothalamus, and other lower brain centers, cause conscious perception of pain. This does not mean that the cerebral cortex has nothing to do with normal pain appreciation. When electrical stimulation of the primary somatosensory cortex area is used, it does cause a human being to perceive mild pain from about 3% of the points that have been stimulated. However, it is believed that the cortex plays an especially important role in interpreting pain quality, even though pain perception might be principally the function of the lower centers, again, our more primal centers. Well, now it's time to talk about capsaicin, and that's how you pronounce it, capsaicin. Capsaicin. Now, this is a chemical that's found in hot peppers, and it's an irritant, and it activates heat receptors in your skin and in your mouth. So when this irritant from hot peppers touches these thermoreceptors, your brain, because again the temperature goes, the stimulus from this capsaicin makes these thermoreceptors detect, quote unquote, an increase in temperature beyond 45 degrees. Your brain now interprets it as pain, which is the reason why if you are a person who eats hot peppers and you don't like something hot, it's because you have very sensitive thermoreceptors in your mouth. Now, I, for one, love me some hot peppers, and I grow all kinds of jalapeno peppers, habaneros. No, I don't grow ghost peppers. They are very difficult to grow in Southern California, and if you are growing them, I'm very jealous of you, but at the same time afraid of trying one. Anyway, they put capsaicin in birdseed because birds don't have the same receptors as we do. And so the idea is if they put capsaicin in birdseed, then other mammals like squirrels, chipmunks, etc. will be less tempted to eat that birdseed. Now, interestingly, apparently capsaicin, if you take chili pepper and sprinkle it in your yard or your garden, maybe because the neighbor's cat keeps coming over and pooping in your garden, if you put that on there, the capsaicin irritates the pads on the cat's feet, and they'd be less likely to go to the bathroom in your garden. Now, that being said, I personally have gone to the 99-cent store and purchased about, I'd say 17 little plastic containers of chili powder, sprinkled it in my garden, kept the neighbor's cat away for maybe 12 hours, and it didn't work. Now, I don't know if it's because it got absorbed in the soil, the sprinklers came on, I don't know. But I'm willing to try it again. Or maybe the neighbor's cat just has very thick pads from being a street cat, out on the road, and not a house cat, and has a lot of calluses on their little pads. I don't know. But capsaicin is used in creams and patches and can be used for controlling the pain of arthritis and shingles. And basically what you do is, you've had that like icy hot cream, you might have used it, but you put it on your skin, and the idea is that when the heat goes up, it overwhelms your receptors so that you are no longer feeling the dull throbbing pain. To put it another way, it's almost like your brain has a different type of pain that is overwhelming it because it's stimulating so many more receptors because you've rubbed it all over your skin, that it has more to contend with this temperature change than the actual pain from the arthritis or shingles. Another way of saying that is it's interfering with substance P, the release of neurotransmitter from the primary or first ordered neuron. It's going to deplete it. It's going to cause the first order neuron to just dump it out, and then there's nothing more to release on the second order neuron. So now let's talk about our natural ways that our brain and spinal cord have the way to suppress pain, or the conscious awareness of pain, stopping the pain signals before they even reach our cerebral cortex. This is our natural analgesia system, pain suppression system. Now again, I want to warn you, it's going to get pretty technical here. The degree to which a person reacts to pain varies tremendously. One person's itch is another person's slice across the skin. Okay, so that's okay. All of us have a threshold for pain. I'm not trying to judge. You shouldn't judge. It just is. This results partly from a capability of the brain itself to suppress input of pain signals to the nervous system by activating a pain control system called an analgesia system. Again, technicalities. The analgesia system is shown in this picture, and it consists of three major components. One, the periaqueductal gray areas, and this is found in the mesencephalon and upper pons. Okay, periaqueductal gray area. Peri, around, aqueduct, around the aqueduct. Shown up here, the top right of the picture, you can see it in gray, around the aqueduct that's in between the third and fourth ventricle. From anatomy, I'll remind you that the third ventricle is bounded by the pons and the medulla. The third ventricle is primarily found housed in the thalamus. And the cerebral aqueduct is also known as the aqueduct of Sylvius. So, we have a region of neurons that are found in this periaqueductal gray area. And I'll remind you, gray means unmyelinated. You should be thinking cell bodies. So, you're seeing the red axon and red cell body that is in the periaqueductal gray area. So that's the first area. Number two, the raphe magnus nucleus and the nucleus reticularis paragigantocellularis. Wow, I just said a whole bunch. Let me just break that down for you. We're talking about cell bodies in the lower pons, upper medulla. So if you look down here with a red axon from our neuronal cell body in the periaqueductal gray area is terminating on a second neuron, it's in the lower region of the pons and upper region of the medulla. Okay, that's where it's just connecting to another neuron, and you're seeing the axon of that orange neuron is going down to the spinal cord. Okay, part 3, a pain inhibitory complex located in the dorsal gray horns of the spinal cord. Okay, follow the orange neuron that is starting in the nucleus raphe magnus in this picture, in the lower region of the pons, upper region of the medulla. Follow it all the way down the spinal cord to the dorsal gray horn, and you're seeing that it's terminating on another short red neuron that's labeled enkephalin neuron. And that's what I mean by the pain inhibitory complex. So we need these three regions. Now, through studies, we know that the analgesia signals can block pain before it is relayed to the brain. And it's blocking pain through that enkephalin neuron. Action potentials are going from the periaqueductal gray matter to the nucleus raphe down to that enkephalin neuron, who's going to release neurotransmitters on that primary neuron, the first neuron conveying information about a pain stimulus before it reaches the second order neuron. And that little short enkephalon neuron is going to interrupt the signal. So it's like the spy. It's going to take the message before the enemy can even detect what the message is. We know this because electrical stimulation, either in the periaqueductal gray area or in the raphe magnus nucleus, can suppress many strong pain signals entering by way of the dorsal spinal roots. Also, stimulation of areas at still higher levels of the brain that excite the periaqueductal gray area have also led to the suppression of pain. These higher levels have been the hypothalamus, just to name a few. I mean, different areas of the hypothalamus. I'll save you the technicalities. Several transmitter substances are involved in the analgesia system. Especially involved are those enkephalins that I just told you about, and serotonin. Many nerve fibers are going to be releasing enkephalin from their endings. And as a result, this is going to block the transmission of substance P from the first order neuron to the second ordered neuron. So again, the enkephalon is believed to cause both the presynaptic and postsynaptic inhibition of incoming type C and type A delta pain fibers where they synapse in the dorsal gray horns. Let me say it again. That enkephalon neuron, when it's stimulated by the higher orders of the brain, the periaqueductal gray area and the nucleus raphe magnus, is interrupting both type C and type A delta fibers. Now in the next slide, I'd like to talk a little bit more about the enkephalins, and these are very short 5 amino acid peptides. They are abbreviated as TGGP-LU or TGGP-MET, and I'll explain that in a little bit. But there's some history to them, so let's move on to that right now. More than 40 years ago, in the 60s and 70s, I hate to say it, but that's back when I was born, it was discovered that injection of minute quantities of morphine either into the periventricular nucleus around the third ventricle or into the periaqueductal gray area, the brainstem, caused an extreme degree of analgesia, pain blocking. In subsequent studies, it has been found that morphine-like agents, mainly opiates, like opium, heroin, morphine, also act at many other points in the analgesia system, including the dorsal gray horns of the spinal cord. Because most drugs that alter excitability of neurons do so by acting on synaptic receptors, it was assumed that the morphine quote-unquote receptors of the analgesia system must be receptors for some morphine-like neurotransmitter that is naturally secreted in the brain. So let me say this in plain English. When scientists first started injecting these opiates into people, either in their brain or in the spinal cord, they had analgesia-like effects. And so the scientists correctly theorized that if these drugs from plants were working in the human, then the human must have an endogenous chemical that was released that would bind to the same receptors that these plant chemicals were binding to. So the research began searching for these morphine-like drugs. About a dozen such opiate-like substances have now been found at different points of the nervous system. All are breakdown products of three main protein molecules, three main ones: pro-opiomelanocortin, or POMC, pro-enkephalin, and pro-dynorphin. Among the more important of these opiate-like substances are beta-endorphin, met-enkephalin, leu-enkephalin, and dynorphin. So, basically, the POMC, pro-opiomelanocortin, is going to be a precursor hormone. Maybe you remember this from your physiology. It's a large protein, and from it can be cut adrenocorticotropin hormone, melanocyte-stimulating hormone, but also cut from it is beta-endorphin. And from the beta-endorphin, or the pro-enkephalin, you can cut smaller pain-relieving molecules like the different enkephalins. Now, the enkephalins, two enkephalins, are found in the brainstem and spinal cord in the portions of the analgesia system. And the beta-endorphin is present in both the hypothalamus and the pituitary gland. The dynorphin is found mainly in the same areas as the enkephalins, but in much lower quantities. So all of these are your natural painkillers, is what I'm trying to say. Although the fine details of the brain's opiate system are not well understood, activation of the analgesia system by nervous signals entering the periaqueductal gray areas, or inactivation of the pain pathways by morphine-like drugs, can almost totally suppress many pain signals entering through peripheral nerves. So let me say this again. Endorphins were discovered almost by accident in the 1970s when scientists were carrying out research on drug addiction. Investigators had wondered for years why the human brain contained receptors for chemicals produced by the poppy plant, and they eventually discovered why. The brain produces its own set of neurochemicals, which are actually far more potent than morphine, opium, and heroin, but they share the same neural receptors with those drugs. The naturally produced brain chemicals, called endorphins and enkephalins, and as I said, a third class, the dynorphins, are released in times of stress. They can make a mangled accident victim as serene as a Buddhist monk. And they can also make an athlete feel great after an extremely vigorous workout. The latter effect is sometimes referred to as the runner's high. And the post-exercise surge in endorphins helps to explain why many exercisers seem to become addicted to their sport. Their workouts become fixes, which mask the pain of everyday living, and even injuries or illnesses can't stop the training process because the athlete is relentlessly searching for endorphin-induced mood elevations. Enkephalins are 5 amino acids containing tyrosine, glycine, phenylalanine, and either leucine or methionine. And that's what I said on the previous slide, and I'm rating here on this slide. They are the smallest of the molecules with pain-killing or opiate activity. Enkephalins are found in the thalamus of the brain and in some parts of the spinal cord that transmit pain impulses. In the spinal cord, enkephalins inhibit painful sensations by reacting with specific receptor sites on the sensory nerve endings. Again, where the first-order neuron is synapsing with a second-order neuron, and that first-order neuron is releasing substance P. That enkephalon neuron that I showed you, that short little red one on the previous slide, is releasing enkephalins. Nerve endings of the central nervous system and the adrenal medulla release these naturally occurring morphine-like substances. Enkephalins bind to opiate receptors and release controlled or undue levels of pain. Leu-enkephalin is an endogenous agonist for the receptors that are stimulated by opiate alkaloids. It has multiple effects on the CNS. Leu-enkephalin and met-enkephalin are both, like I said, are natural pain killers. Met-enkephalin in particular is involved in a phenomenon associated with modulated pain perception, regulation of memory and emotional conditions, food, and liquid consumption. It also has an impact on digestive system, motility, gastric, as well as pancreatic secretion, etc. So the met-enkephalin is that feel-good drug that is released when you eat food of sweet rewards. You know, your comfort food? That's what is stimulating the release of the met-enkephalin. Now, again, in addition to the runner's high and eating foods that give you comfort, we also see surges of these natural painkillers in both the mom and baby during birth, because for both it's painful. We see this occur in death as part of the reason why people, when they have near-death experiences, they'll tell you that they feel no pain even though Mr. Whitey, great white shark, bit off their leg. But it's also important to note that in people who are taking exogenous morphine and they immediately withdraw from it, they will have withdrawal symptoms that are so intense in pain. It's because the morphine is occupying, I should say, these receptors, and so the natural enkephalins, it's like a negative feedback effect, are not being released. So if the patient stops morphine cold turkey, then their own body is not releasing their own painkillers. And so the smallest stimulus can be perceived as very, very painful on the scale of 9 to 10. So the drug naloxone is a way of basically working them down from that morphine so they don't have extreme withdrawal. Now the last part that I want to follow up with on this slide is THC, tetrahydrocannabinol, which is basically marijuana. Now THC, the substance found in marijuana, causes a release of enkephalins, a natural release of enkephalins. So you get this analgesic effect, but you also get that euphoric effect like taking opium or heroin. So it's kind of a mixed event because they know that some of that a person can become addicted to, that euphoric reaction. And if you ask someone who's doing marijuana, ask them, are you addicted to it? They'll say, no, I can quit any time. And it doesn't always work. But we know on a scientific level that that drug, THC, actually does stimulate the natural enkephalin release. And there's that kind of pain relief and euphoria experience that they will report. So there is a relationship between enkephalin release, and addiction. So those who are saying that THC, the substance found in marijuana, is not addictive, the science would suggest otherwise. Now, to get back to your chapter and talking about problems with pain, your chapter talks about pain threshold, which is basically the minimum stimulus that can be perceived as painful. There's also pain tolerance. That means how much can you tolerate before you're going to pass out. And usually this is on a scale of 1 to 9. When they first did these studies, it was actually on military personnel. And since then, in a clinical setting, well, we just round up to a scale of 1 to 10. We can further break down pain as nociceptive pain, where the nociceptors are aberrantly activated, or neuropathic pain, or neuropathy. And this can result from either direct injury to neurons or dysfunction of sensory neurons, specifically the axons. Now we can take it a step further and say, are these neurons oversensitive? In which case we say neuropic hyperalgesia. Algesia means more sensitive to pain. And this person with this neuropic hyperalgesia would be more sensitive to pain. And the classic example of that is tic douloureux. Now, tic douloureux is basically this neuropic hyperalgesia, and I'd like to tell you a little bit about it. It occurs in some people over one side of their face in the sensory area, and it's usually because of cranial nerve 5 or 9. So it's either due to trigeminal neuralgia or glossopharyngeal neuralgia. The pain feels like sudden electrical shocks, and it may appear for only a few seconds at a time or may be almost continuous. Often it is set off by exceedingly sensitive trigger areas on the surface of the face, maybe in the mouth or inside the throat, almost always by a mechanoreceptive stimulus, meaning something touching rather than a pain stimulus like something they ate, for example. For instance, when the patient swallows a bolus of food, as the food touches a tonsil, it might set off a severe lancinating pain in the mandibular portion of the fifth nerve, trigeminal nerve. The pain of tic douloureux can usually be blocked by surgically cutting the peripheral nerve from the hypersensitive area. The sensory portion of the trigeminal nerve is often sectioned immediately inside the cranium, where the motor and sensory roots of this nerve would separate from each other. So that means you're sparing the motor roots from the sensory part. So the person won't have paralysis of their facial muscles, specifically the muscles of mastication, which cranial nerve 5 would control. Now, the opposite extreme would be neuropathic analgesia, which can happen in diabetic neuropathy, which we've already discussed. In this case, there is absence of pain from stimuli that most certainly should be painful. And again, to remind you, pain is supposed to be protective, and we store memories on it. We're supposed to learn from it, sort of like saying, well, I'll never do that again. Or when I saw that nice brown fuzzy creature, I thought it would be something that I could play with. But when it mauled me almost to death, I realize now that that large bear is something that I should avoid. Neuropic analgesia, as I described in our just last week's lecture, can happen in diabetic neuropathy because of the high blood glucose levels, leading to vascular changes, and neurons need blood vessels too for the delivery of nutrients. And without these nutrients being delivered or oxygen being delivered, the neurons become dysfunctional. They do not relay the sensation of pain. And I was telling you about new shoes with different pressure points. The person is not aware of them, and therefore the blisters turn into ulcers, and they are poor for healing. There isn't adequate blood flow. So neuropathy that leads to inappropriate analgesia is not okay. That's the opposite of being hypersensitive to pain. Now I'd like to conclude our talk on headache. Okay. Headaches are a type of pain referred to the surface of the head from deep head structures. Some headaches result from pains inside the cranium, but others result from pain arising outside the cranium, such as from the nasal sinuses, for example. So I'd like to first focus on headache of intracranial origin. Now, first of all, I'd like to say the brain tissues themselves are almost totally insensitive to pain. Even cutting or electrically stimulating the sensory areas of the cerebral cortex only occasionally cause pain. Instead, these sensations cause prickly types of paresthesias on the area of the body represented by the portion of the sensory cortex stimulated. And that goes back to the experiments that I was telling you about, Wilder-Penfield, with mapping the homunculus. Conversely, tugging on the venous sinuses around the brain, damaging the tentorium, which is a piece of the dura mater, or stretching the dura at the base of the brain can cause intense pain that is recognized as headache. Also, almost any type of traumatizing, crushing, or stretching stimulus of the blood vessels of the meninges can cause headache. An especially sensitive structure is the middle meningeal artery, and neurosurgeons are careful to anesthetize this artery, specifically when performing brain operations under local anesthesia. Types of intracranial headache can include headache of meningitis. One of the most severe headaches of all is that resulting from meningitis, which causes inflammation of all the meninges, the dura mater, the arachnoid, the pia mater, including the sensitive areas of the dura and the sensitive areas of the venous sinuses. Such intense damage can cause extreme headache and pain referred over the entire head. Headache can also occur by low cerebral spinal fluid, in which case the brain is not as buoyant as it should be. It sinks to a more inferior position, and this pulls the meninges taut and again can stimulate the receptors that are in the meninges. Migraine headache. Migraine headache is a special type of headache that may result from abnormal vascular phenomena, although the exact mechanism is unknown. Migraine headaches often begin with various prodromal sensations, such as nausea, loss of vision in part of one field, visual aura where they see spots, and other types of sensory hallucinations. So there's like a preemptive warning. Alcoholic headache. As many people have experienced, a headache often follows excessive alcohol consumption. Consumption. It is likely that alcohol, because it is toxic to tissues, of course, directly irritates the meninges, and it can directly irritate the meninges because it can cross the blood-brain barrier and therefore causes the intracranial pain. The dehydration, of course, also results from alcohol intake because alcohol inhibits the release of ADH. And so that can lead to a loss of cerebral spinal fluid and pulling on the meninges. So the hangover from an alcoholic binge, it's usually hydration that is the issue. And hydrating can attenuate the pain of an alcoholic headache, but it does not abolish the headache and other symptoms of the hangover. So you can drink your water while you're drinking your wine, and that might attenuate your hangover for the next day, but it won't abolish it. Now, extracranial headaches. These can be headaches that arise from muscle spasm. Emotional tension often causes many of the muscles of the head, especially the muscles attached to the scalp and neck muscles, to become spastic, held in contraction, and it is postulated that this is one of the common causes of headache. The pain of the spastic head muscles supposedly is referred to the overlying areas of the head and gives one the same type of headache as intracranial lesions do. Again, it's like a referred pain. Headache caused by irritation of nasal and accessory nasal structures. Have you ever been in a car with someone wearing perfume that just irritated your nasal cavity and it led to a headache? The mucous membranes of your nose and nasal sinuses are sensitive to pain, but not intensely so. Nonetheless, infection or other irritative processes in widespread areas of your nasal structures can summate and cause headache that is referred to behind the eyes, or in the case of frontal sinus infection, to the frontal surfaces of your forehead and scalp. So that person wearing the annoying perfume, you get the headache and you squeeze your eyes together, that's referred pain to behind your orbital area. Headache, of course, can be caused by eye disorders. Difficulty in focusing one's eyes clearly can cause excessive contraction of the eye ciliary muscles in an attempt to get your vision cleared. And even though these muscles are extremely small, it is believed that constant contraction of them can cause retroorbital headache, meaning even behind the orbits. So, when you're studying, it is suggested that you look up every 20 minutes, look 20 feet away, and blink 20 times over so that you're not holding your intrinsic eye muscles too extensively, which can lead to eye strain, vision problems, and then headache. So every 20 minutes, a good optometrist will tell you the 20-20-20 rule. Every 20 minutes, look 20 feet away and blink 20 times. All right, I hope you enjoyed your pain pathways and discriminative touch pathways and what can cause the perception of pain and how we block pain. Next week is our last lecture.